All right, well, thanks everybody for coming. We are proceeding with the next instance in the seminar, pre-kickoff seminar series, going through the different topics in the Institute. And this one will be by Amy Orsborn, Eli Schleiserman, and Maria Bedarlot about neuroscience applications. So please go ahead, Amy.
Great. So our group is broadly interested in neuroscience, which, at a high level is really trying to understand how our brains are controlling our behaviors, and then also trying to develop therapies to treat neurological disorders. And the main reason this is hard is because brains are really, really big and complicated. So for instance, the human brain has billions of neurons, and they have trillions of connections between them that allow neurons to pass messages from one to another. And so what this means is that computations that the brain is performing are distributed across a large network within the brain. And at the end of the day, the computations are happening across multiple scales. So each individual neuron within a brain area is performing a computation, but it's not performing that computation in isolation. We also have to think about how it's computing things when it interacts with other neurons across many parts of the brain. And this is going to be true for pretty much any behavior. So if you think about reaching out and grabbing a cup, it's not just involving the motor areas as I'm moving my arm, but we also have visual areas, sensory areas, seeing the glass, understanding when I'm making contact, and so on. And so this is also then going to be true for most any neurological disorder. They all involve some disruption to this network within the brain. And so to take a quick step back, let's talk a little bit about neurons themselves that are the main computational element within the brain and how they are performing computation and sending messages within this network that they're embedded in. So one of the really interesting things about neurons is that they have a mixture of electrical and chemical signaling. So neurons are cells, and they have an interesting structure where they have dendrites, which basically are where they receive messages from other neurons. And then they have axons, which are where they send their message to other neurons downstream. And so what happens is that in a particular neuron, they receive chemical signals from other neurons at their dendrites. They translate those into electrical activity within the cell. And when they receive the right combination of messages, they generate an action potential. That's an electrical signal, and that's a current that travels down the axon and then leads to a chemical release at the end of that cell that then is passed on to the other neurons. And so what that actually looks like is that the action potential, that electrical current, travels down and leads to release of what we call neurotransmitters, but basically just chemicals that then those chemicals travel down to other neurons, and they have receptors on them, on their dendrites, that take that chemical in and then turn it into a change in electrical voltage within the cell. And so given that we have this interesting mix of electrical and chemical activity, there are many different ways that we can measure activity in the brain. So obviously one is to measure electrical activity. We can detect currents or voltages, and we can do that inside or outside of neurons to gain inferences about those action potentials, for instance, that they're sending. And then we can also look at chemical information. So you can detect the changes in the ion flows inside and into or out of a neuron. And you can also detect those neurotransmitters that are released. And then, of course, because neurons are cells, they're also metabolic indicators of neural activity. So for instance, blood flows more to areas where neurons are firing quite a bit because they're consuming energy. So if you've ever heard of functional magnetic resonance imaging, that's actually what those scans are detecting. They're looking at the changes in blood flow in the brain rather than the electrical or chemical activity of neurons directly. And in addition, we can make all of those measurements at lots of different scales. Within any modality, you could try to make really precise measurements of individual neurons, or you could make measurements that capture activity of kind of groups of neurons together. And so one of the reasons that neuroscience is really kind of in an interesting position to start to benefit from artificial intelligence is that there's been kind of a revolution in all of these different technologies to measure neural activity. And a lot of this was driven by the BRAIN Initiative, which you might have heard of. It's a large initiative started by Barack Obama to really push developments in neurotechnology. And so all sorts of new advanced electrodes, magnetic resonance imaging approaches, optical ultrasound, kind of combinations of different methods, et cetera, have been developed and improved over time because of this initiative. And so to give just some quick examples, if we look at electrical activity, so which is a really common approach for looking at neural activity, the historical approaches to this have been relatively simple. You basically put a very, very tiny wire inside of the brain. And when you get that wire close enough to neurons, you can pick up those action potentials. So if all of these different gray dots here represent different neurons, that wire is ultimately only going to be able to pick up action potentials from maybe a couple of neurons right nearby. And you could put in at most a couple of these wires together at a time. Now, over using advanced electronics, we've shifted towards that wire being replaced now with a sensor that has 400 or more different sensing pads on it, really closely spaced together. And so we can make now, instead of recording from just a couple of neurons, we can record many, many neurons all around that single wire that we're putting in the brain. By using advanced material science, these probes are getting thinner, smaller, so we can pack them more densely, and we can also, that increases their biocompatibility, so you can make these measurements for longer and longer periods of time in the brain. And so recently, for instance, I collaborated with a group of people who developed some really high density sensors that you could put on the surface of the brain that could measure from 1,000 electrodes within just a 1 centimeter squared area. And that within a human brain is kind of a drop in an ocean, right? The brain is much bigger than that. But here's just a quick video to give you a sense of what we start to pick up when we can make these really high density measurements. So this is activity as a monkey is watching just a simple video played to them. It's a very simple behavior, but you can see that there's really complex patterns of activity across the brain that we can resolve with this. In addition, there's been huge advances in optical approaches. Maria will talk a little bit more about this in a minute, but optical techniques are driven by sensors where we can actually have neurons change in their fluorescence intensity as calcium, for instance, or other indicators of activity in the cell are changing. And so what you're seeing in this video, let me restart it, is a big picture, zoomed out image of mouse cortex, and those changes in flashes of light are neurons firing. And then when you look at each individual neuron with high resolution imaging, you can then get a time series of how activity is changing in that neuron over time. And so these optical approaches have kind of really allowed a huge advance in the resolution and specificity of the measurements we can make, because you can also genetically target, for instance, these indicators to show up in certain kinds of cells, but we can also do this over much larger volumes as well. And so those are just a couple examples of electrical and optical approaches. There's all sorts of other really cool sensors that have come out of the Brain Initiative, but focusing on those because that's the main thing that I and Maria work on. And then in addition, ultimately, we care a lot about relating that brain activity back to behaviors. And one of the things that's been really interesting in the last couple of years is kind of a huge boom in using computer vision and other advanced approaches to monitor behaviors at a new level of detail. And so this is just a snapshot of a paper that recently was published where they've developed these large arenas, and you can have many, many cameras and then track movements of monkeys in this case, but you can as they move around this large volume and do really complex, rich tasks. And so the ways that we're tracking behavior are also really expanding very, very rapidly and leading to large piles of data. And so in general, all of our data is getting bigger, but it's worth kind of talking for a second about the way in which they're getting bigger. So in neuroscience, we care a lot about the temporal and spatial resolution of the measurements, in addition to the coverage, right? Because we're always struggling with this issue of making measurements from a really, really large network. And so to take a quick snapshot of previous existing technologies, things like functional magnetic resonance imaging are really great at getting spatial coverage. You can take scans of the whole brain to understand this sort of large distributed computation, but you have, because you're measuring metabolic indicators, blood flow, the signal has really, really low temporal resolution. And you also have pretty low spatial resolution because you're not looking at individual neurons, you're kind of looking at many, many neurons together. On the other end of the spectrum, we have things like those electrodes that I talked about, the wires that you can put in the brain. And you can maybe make a group of wires like this electrode array that I've shown here. And when you do that, you get much, much better temporal and spatial resolution. But you don't have a lot of spatial coverage because you're kind of only getting to measure from a tiny patch within the large brain. And these recordings tend not to last for very long because of biocompatibility issues. And so you don't get to record for very long. But with some of these advanced electrophysiology techniques, like I mentioned before, these really high density thin probes, what these technologies are doing is sort of taking that high temporal spatial resolution of these measurements, but then increasing the coverage in space and time that we get. And so this is really leading to kind of giant piles of data that neuroscientists certainly need help with understanding how to analyze. And in fact, there's a really nice analysis that showed that the number of simultaneously recorded neurons has sort of a equivalent of Moore's law. If you look at sort of how this is evolving over time, that it has doubled roughly every 7 years. If you look back from 1960, I haven't seen this plot gone further out. I suspect the rate is increasing even a little bit more. And then the other kind of important thing that I'll just comment on in terms of like big picture data things is that our data is also becoming multimodal. So as you might sort of guess from some of these examples that I've shown you, given the scale of the measurements that we're trying to make, no recording method really is sort of optimal for solving all of our problems. So they all have pros and cons. For instance, imaging gives us this really dense spatial sampling, but it has comparatively low temporal resolution. If we do things like electrophysiology, we get the inverse, where we have low spatial sampling, but really high temporal resolution. And so there's always going to be, to some extent, many trade-offs in this spectrum. And so that's led a lot of people to think about multimodal approaches and multi-scale sampling. So this video is one example of that. That's an approach called a mesoscope, but these videos play a little fast for trying to explain them. But what you see here is sort of a low resolution image of all the whole hemisphere of a mouse brain. And then what they've done is, based on that macroscopic image, pick smaller regions of interest that they then want to image in high resolution. And so this mixture of of scales of measurement has become really powerful for trying to grapple with some of the measurement trade-offs that we have to make. And of course, as this audience definitely knows, more data, doesn't necessarily solve all the problems. It also creates other problems. And so there's a big need for kind of increasing the computational efficiency of data processing in neuroscience. And in particular, there's a lot of need for real-time processing. And I'll talk a little bit more about that in a second. But particularly when you try to think about therapies, some things that you may have heard about before, things like brain-machine interfaces or stimulators, like brain stimulation for Parkinson's, there's a need to sort of process neural activity in real time in order to guide these different therapies. And so with that, I'll transition to talking a little bit about some of the, zooming in a little bit more on kind of some of the things that we're thinking about in the context of A3D3 and my work. And then I'll transition to Maria, who will tell you a little bit more about some of the things she's working on. But I'm particularly interested in this challenge of sort of real-time, or what we call closed-loop manipulations, for studying brain behavior and also for thinking about therapies. So just some examples of how this is useful in neuroscience. Like I mentioned, the brain is this big network, and there's connections between neurons. One of the most interesting things is that these connections are dynamic, so they can change over time as you learn different things. And so, for instance, there are applications where you might want to induce that plasticity in an artificial way, where maybe we record activity from one neuron, and then based on when this neuron is firing, we stimulate the activity to drive activity in another neuron. And if you do this in a really precise time-locked way, you can actually cause those two neurons to increase the strength in their connection. Another example would be to stimulate the brain based on detected brain states, and then study how that impacts the behavior. And I'll talk a little bit more about this in a second. So, but the idea is that you could then, this provides a causal intervention to try to understand how a particular pattern of neural activity or state in the brain is related to behaviors. And then in addition, we have sort of this interesting challenge in neuroscience where, behaviors or even like visual stimuli in the world are really complicated. If you try to think about a parametric way to describe all of the different things that we see, it's a really, really long list. And so trying to understand how neurons are representing that really complex space is a big, even just statistical challenge. And so one approach that people are using to get around this is actually closed loop manipulations, where you decide what stimuli to show a particular subject based on the way that that neuron is responding. And that allows you to basically do active learning and sample the space of what this neuron might care about in the visual field in a more effective way than just brute force sort of showing it, red, green, blue dots and so on. And so in that space of kind of closed loop manipulations, the application that we're focusing on for D3 is thinking about doing closed loop stimulation based on brain states or detected events in the brain to understand how those events relate to behavior. And the particular example that we're thinking about is something called a sleep spindle. So if you measure this signal called a local field potential, which captures aggregate activity of many neurons, you'll often see these really interesting brief events of these somewhat high frequency oscillations. And that is called a spindle, and it's pretty rare. And it primarily occurs actually while you're sleeping, or maybe while you're kind of taking a break. And it seems like these spindles are contribute in some way to learning. But most of that's just been gained through kind of correlational assessments. And there are a few experiments where people have been able to detect that a spindle has started and then stimulate, for instance, to disrupt it so that you don't allow that spindle to happen. And when you do that, you find, in fact, that you can disrupt the subject's ability to learn a particular behavior. And so this further reinforces this idea that it's important in learning. But our goal here is, instead of kind of detect that one has started and then shut it down, is could we actually predict that a spindle is going to occur before it has happened, and then stimulate to completely prevent the spindles from happening? And so that's the kind of thing that we think that A3D3 could be really useful for. And in particular, because the goal ultimately would be to actually package this thing on an animal's head, right? So that I work with monkeys, you could have a monkey that was sleeping in their cage, you detect spindles in real time, and then stimulate. And so this is just a quick, I'm not going to sort of explain the details because it's not immediately relevant, but this is an example of, the kind of implants that we package and put on a monkey's head. The diameter of this is about 3 centimeters to give you a sense of the scale. But, and then we're making measurements from, large areas, and our goal would be that you could have ultimately packaged hardware that we could add to somewhere in this stack that the animal could wear, or potentially to wirelessly transmit to another processing unit. And so with that, I will wrap up. I've probably taken up a decent amount of time, but I'll transition now to Maria. We'll talk a little bit more about some of her applications that for A3D3.
Okay, great. Thank you, Amy. Let me turn on my video and I'll share my screen here. Okay. All right, so yeah, and thinking about the kind of work that I do and Amy does, I would steal a little term she used earlier and call it sort of multimodal neuroscience. And the reason I would say that is that we have two sort of broad general goals. And I think a good subset of neuroscientists could be described this way, where, we want to understand how natural neural systems actually work. How does it work in our system? For example, how do humans interact with the world outside of them and act upon the world outside of them? And one of the reasons that we want to understand that is because we are also biomedical engineers, and we're looking to build artificial neural systems to replace these natural neural systems when they break down due to injuries such as stroke or incurable diseases such as ALS. And so just to give a concrete example of these two types of processes, an example of a natural neural system would be visual processing. And in particular, I'm going to talk a little bit later about hierarchical visual processing. The sort of parallel artificial system would be artificial sensation for vision. So what does it mean to have artificial sensation, or to want artificial sensation? Well, in a nice study that just came out recently, it's all over the news right now, provides a very good example. So there is a woman who has been blind for a long time, and she had one of these Utah arrays. This array that Amy showed is so famous, it has its own name. It's a Utah array developed at the University of Utah. And it is small, so 4 by 4 millimeters with 100 channels. And they implanted it in the visual cortex of this woman. So it's the part of the brain that responds most strongly to visual inputs. And so the nice thing about these neurons that we talked about, we said that within a neuron, you transmit information electrically. And so what that lets us do then is, if we're passing tiny little electrical currents through the tips of each of these electrodes, we can actually affect what neurons are doing. And that lets us implement this artificial sensation. So you pass a tiny electrical current through one of these electrodes, you can activate a neuron, and that leads to a percept, right? So if you do that in a human being or a monkey, and you ask them what they saw, indirectly in the monkey, of course, they would say they saw a tiny little point of light. Okay, and the nice thing is that the brain is thankfully has a very high level of spatial organization. So if you stimulate on one part of this array versus on the other part of this array, then you're going to elicit different percepts. And then you can start to think about stimulating across multiple points on this array to elicit more complex percepts. So one example that was demonstrated in this paper is that you can elicit perception of these distinct characters looking like an O, an L, and a cross by using different patterns of multi-channel stimulation across this electrode. This woman also could learn to do things like localized borders between dark and white. That's a big function in the visual cortex and one of the very early steps in object identification. And also she could localize, for example, the position of white squares. All right, so, so obviously one of the important things in vision is perception, right? So just like I described, you can perceive what is in the outside world. But sorry, I'm just looking at my time to make sure I'm not going to go over. But it's more complicated than that, right? Because we exist not only as passive vessels in the world, we're also acting upon the world. And so perception is actually intimately integrated with behavior and they affect one another. So one example is that you could, for example, have the exact same type of visual input that is self versus externally generated. So you can imagine looking outside the window, like I'm doing right now, and seeing a bird on a tree. And if I turn my head, that bird is going to move across my visual field. But also, if the bird were to, if I kept my head straight and the bird were to take off and fly, I could get theoretically the exact same visual percept of the bird moving across the visual field. So, one of the roles of our nervous systems is to differentiate between self-generated perceptions and externally generated perceptions, because it's important to know what you're doing. And then another complication that we've sort of skimmed over here is that neural responses to things like vision, so basic sensory inputs, it changes based on what else you're doing. So even going from being a little bit sleepy to more alert, even changes like from standing still to starting to walk through the world, then your responses to the exact same visual input is going to be very different. And so this leads to a really complicated problem if we're thinking about building these artificial neural systems. How do you take all of these parameters into account? And can we even? Just a little bit more about how complicated the sort of natural neural system is and describing the task that we are posed with. When we think about hierarchical visual processing, it's saying, okay, you have different areas of the brain that are working together to figure out what am I looking at. So I like this example from this nice classic book, The Principles of Neuroscience, where you're looking at a very complicated picture with lots of features. And one of the main features is, okay, we have a bunch of horses. Well, what does your visual system actually have to do? So first of all, one of the main tasks that it does is pick out little oriented bars, oriented areas. So orientation, direction. If I'm looking here, where is the border? If I'm looking here, where is the border? If I'm looking here, where is the border? It has to figure that out across the entire picture. And then also processes things like color in different parts of space and contrast, how fuzzy or not fuzzy is it? And so on and so forth. Okay, and so this is the first stage of processing, and then we have to extract information. So now that I have a bunch of orientations in different locations in this visual field, how do I put it together to then come up with this sort of coherent object where I just say horse, whether I'm looking at this horse or that horse, whether the horse is upside down, and so on and so forth. So that's still an open question. How does this exactly happen, and how does it interact with behavior? And because this is such a complicated process, it happens across many different parts of the brain. And so ideally what we have moved to trying to do, what the latest technology has let us do is actually record from many different parts of the brain at once in order to start to try to answer that question. So this is an example of a mesoscope, which is similar to the one that Amy showed earlier and is what we have in my lab at Purdue. And we have these very nice mice, and they're not normal mice, they're a special type of mouse that are transgenic. So they have been bred, let's say their grandmother's grandmother's grandmother as an embryo was transfected with a virus that led to neurons in the brain of the mice expressing a fluorescent particle that changes its intensity when the neuron is active. And so we use a laser to excite neurons within the brain. And so we have access to neurons within the brain, I should say, because we take off part of the skull and we implant this cranial window. So now it's sitting right on the surface of the brain. So we have access to neurons within the first, say, 500 micrometers of the brain, and that is about half of the cortex, which we think is sort of like the most advanced and most lately developed part of the brain. And so you can image from this mouse while it's behaving or engaged in some task or viewing images like the horse pictures we saw before, and then ask what different parts of the brain are doing. So this is just an example of a picture that we took recently. So the microscope has an electrically tunable lens, so we can simultaneously record from multiple layers within the cortex. The cortex has a sophisticated organization where, at different depths, it's doing slightly different things. So we can record from multiple depths and at the same time maybe get around 3,000 neurons across these two depths and spanning about a 1.2 by 1.9 millimeter field of view. And so within this area, you're getting many different parts of the brain. And so if we zoom in, I'm just taking one of these planes right here and imagine zooming in. One of the issues we have, of course, is if you're going to sample from such a large area, your single neuron definition is not ideal. So, this is a neuron here, these bunches of pixels. Okay, so here are some sample traces. You say, okay, this is a neuron. I'm going to take the average activity within this boundary that I have defined as the neural cell body, and then look at how it changes over time. And so here's just an example of 50 of these recorded neurons and the dynamics of the neural activity as a function of time. And one thing to note is that, the nice thing I should say about the mesoscopic 2-photon imaging system is that we have this huge field of view and get so many neurons. But unfortunately, the problem you end up with is that you have so many independent variables, if we're thinking about each neuron as an independent variable, and so few relative samples. So we have a sampling rate of about 3.9 hertz per pixel within this plane. And so I'm ending up with 3,000 neurons and only 9,000 samples, which is about 40 minutes. And so there are technical details I won't get into, but there are reasons that you don't want to or even can't record for the same neuron for much longer than that. And so this leads to a problem where we have sort of poor time resolution in the system. And really what it lets us do is look more for sort of spatial code for sensation and sensory processing. And also, it really leads to the importance of post-processing. So how do you do dimensionality reduction in order to keep information from the entire population of neurons, so you're not throwing anything away? And how, for example, would I segment the field so I can isolate different brain areas and ask what they're doing at distinct points in time? And I'm running long, but and this will let us do something, using this system and combining it with behavior. So for example, we can put mice in a virtual reality system and then track their movements. And then you can have a closed loop system where you can manipulate the visual input that they're actually seeing based on their movements and sort of cause surprises, suddenly switch the visual scene, or even dissociate what they're seeing from what they're actually doing, to better understand how the brain differentially represents this sort of externally imposed vision versus self-generated visual flow. And that's a sort of technique that my lab is looking at. And this is just a movie showing we're doing three-dimensional reconstruction of animal behavior in time. And then, once we have a better understanding of how these natural systems work, we really are working towards improving our artificial neural systems. One of the big questions is, so we're looking to stimulate and activate different patterns of neural activity in the brain to sort of encode information within the brain. But what do you do? What patterns should you use? How should they vary in time to evoke complex behavior? So we have a technique where we're combining 2-photon imaging with electrical stimulation by having a chronically implanted electrode under a glass imaging window, and then looking at neural activity as a function of both time and stimulation parameters. And also we have an in-cage behavioral training system. And this is something where real-time processing and closed loop stimulation becomes very important, because we're trying to provide the mouse information as he's moving around his home cage. But as I said, the neural activity, the ongoing neural activity, strongly depends on what the mouse is doing. And also, similarly, the evoked neural activity by stimulation depends on what the mouse is doing. And so I think this is a big area where integrating with other people in A3, D3 will help us figure out the right thing to do. Now, I know I've gone kind of long, Eli, so I apologize, and I will hand over the floor.
You're muted.
Right, all over. Thanks, Amy and Maria. So Amy and Maria talked about experimental approaches and how data is generated. My lab is working on methods that we can introduce for these questions of both addressing neural recordings and also addressing behavioral data, how eventually we do correlation between these different types of data. I should say in the beginning that interpretation of data for nervous system, for neural systems, is kind of like at the initial stage due to, as was described by Amy, because of still developing methodology to gather data and also behavioral experiments. And also because this data is quite different. So these are kind of like continuous spatial temporal signals that need to be processed, and typically need to be processed also with a high resolution in space and high resolution in time. So our main goal so far is in data interpretation for neural systems. And we could think of data interpretation in terms of three major kind of aims and tasks. With these multi-neural recordings, we would like to do first kind of like a data organization. So when data is being recorded, we would like to understand what are the clusters in this data, and do they correspond to kind of like different types of patterns, as was described in previous presentations. Then the next interesting question is to consider classifications, so to associate these patterns, these clusters, with particular types of first neural activity, and then to correlate it with also behavioral outcomes with natural stimuli, whether these are different classes or kind of like a superposition of classes that correspond to behavior. The third direction is to learn from the recordings what is the structural formation of the network. how neurons eventually form networks such that they are processing and controlling the behavior. And these three aspects, we identify them as the main aspects for our team in A3D3 to study in collaboration with other teams. And it works well with real-time processing. This is our challenge, and also high-resolution processing. And I'm already using a version of almost on the way to be a final icon for our institute. Beyond the data interpretation applications, I just wanted to mention that this is a really useful exercise, not just to organize the data and kind of like to understand what the data tells us, but to transfer from data to system understanding and applications. And these are the examples that kind of like the data interpretation will facilitate. So from data, we could go to modeling neural dynamics, then to model how these systems actually work and produce control behavior. And I'm giving here several examples that we already started to work with very simple organisms. And also control, so closed loop applications, then taking the lessons that we learned from data processing and then targeting them towards control. Both control, which is a neuromodulation control, so it's like using these Utah arrays or even bigger arrays in terms of stimulating neurons at the right time, and also structure control, how we can rewire maybe the network such that it will be, like, for example, repaired from a damage. But this is more futuristic in terms of the data approaches. All right, so in terms of clustering multidimensional time series, I wanted to survey quickly what are the classical approaches. And the classical approaches are mostly in terms of dimension reduction. So the goal is to take as much data as possible and then to see whether there is some low dimensional kind of like representation of the data. So we have introduced several approaches, and there are other approaches as well, as compressed sensing and so on. Most of these approaches are saying that it is possible indeed to find a low dimensional representation within neural recordings, which is very interesting and also prospective for applications. So we could represent the data from multiple channels or multiple kind of neurons being gathered together into clusters in a low dimensional space. And here you can see an example of, for example, recordings from the olfactory system, where each representation, each cluster will correspond to a different scent that is given to an animal. The main lesson from these classical machine learning methods is that the more neurons, the better is the clustering in this low dimensional space. So it's almost a little bit non-intuitive that you would need much more data in order to kind of derive at a better, a cleaner low dimensional representation. But these are all the lessons. It also hints that if you have the right kind of like signal that you are working with, this would be an easier task than just like a sampling kind of like random signal in terms of like, would you work with the spike data or like imaging data of the neurons or all of them together. So if you are able indeed to find this representation that gives you also low dimensional space, this becomes a powerful tool. So over the last years, we are developing more modern deep learning approaches exactly to find this low dimensional representation or latent space representation of neural activity. We are mostly using, as you can see here on the right, the setup that, that is called encoder-decoder, where the signal is being given as an input, and then it is also given as an output in the decoder stage, and then it is being compared to each other. So the task is whether to reconstruct the same signal through the network or to predict what will happen next to the signal. And these tasks turn out to be very informative in terms of building latent representation. And this latent representation can be used then with low dimensional methods in terms of processing and building the classification space or clustering space. So in terms of clustering multidimensional time series, here are several examples from our work that we look forward to apply to neural data that we will be considering, and also other applications, and kind of like to merge them with different applications in the center. Is from a behavioral standpoint, we see that the encoder-decoder applications are very powerful. So for example, we can look at the behavioral data, and as Amy and Maria described, behavioral data is important because eventually they are correlated with neural data. So in this example, actions of human movements are being tracked, so the joints are being tracked, and we would like to, from a sequence of human movements, we would like to classify them into different actions, so into clusters, and then to use classifiers to classify them into different actions. What we see is that these unsupervised methods in terms of encoder-decoder are very useful in terms of clustering, and eventually the network will learn the representation by self-organization. So you won't need to impose too many conditions or no conditions at all, such that the network will cluster those actions that are close to each other into like close by clusters, and those that are far from each other to different clusters at all. More than that, there are also examples that in terms of neural data, it can be also clustered in terms of those clusters that correspond to different activities. And this is kind of like a state of the art currently in neuroscience in terms of considering similar encoder-decoder networks and then to learn the representation. Currently, it was applied to spiking data, and a work in progress with Amy, we are applying it to multi-channel ECOG data, and we are seeing promise in such approaches. Eventually, our goal would be to combine these two different types of data, behavioral and neural, towards kind of like a single model that is working in tandem. I just wanted to mention that this task of prediction and reconstruction is already useful in terms of denoising the data, in terms of reconstructing some channels that are maybe on a slower scale, in terms of tracking movement, for example, like helping if you're both, so channel reconstruction would be kind of completing spatial information, and tracking would be completing temporal information. All these are already included in considering the prediction and the reconstruction task, and very useful already for applications in neural data. All of them need to be done in real time because data is being recorded, and we would like to see what is happening as it is being recorded. And the anomaly detection, as Amy was mentioning, for example, detecting a spindle in neural recordings, that would be something that would be out of the prediction and the reconstruction kind of like known to the model. We are thinking of it as applicable to kind of then to see what are the deviations from the representation that the model learned towards kind of like detecting different anomalies that will correspond to spindles and other interesting features in the data that would appear kind of like as rare events. So we are also working from recordings to structures. So I just wanted to mention here kind of like we are constructing eventually statistical models in terms of how do you take recordings and then form back a model that will explain what is the flow of information in the system, how the neurons need to be connected such that they will facilitate such behavior. And the last type of methods that I wanted to mention is classification of multidimensional time series, which means that after the data was clustered, a really interesting question is then like to see whether these clusters correspond to behaviors, and if indeed they correspond to different behaviors, functionalities, to label them, to associate a name with each cluster. So this requires labeling, and typically with the neural data, labeling is really hard because you would need to relabel the data every time. Also with behavioral data, it would need quite a lot of labeling. For example, when you are going from one organism to another organism, this will completely change the behavior. So all these aspects are very promising, but they require quite a lot of labels and strong supervision. So what we are working on is to go from unsupervised, so from clusters, to kind of like a area in between full supervised to semi-supervised or active learning approaches in terms of associating entities to clusters. Here is an example system that we are working on. In the system, we are proposing that encoder-decoder structures could work as unsupervised organization of data, and then we are seeing that active learning components could be attached such they work together with the unsupervised approach here on the left, such that they help to annotate some entities in the different clusters, and then to both cause the clusters to kind of like be more far apart if they are different enough, and also to associate labels to these clusters. So this summarizes the different methods that we are studying, and we are seeing that this approach could kind of be really the bridge between neural recordings and interpreting neural recordings, and also associating them with behavioral data. So this is my last slide, and next we will take questions.
Thank you very much, everybody. That's all three of you. That's super interesting. Totally unfamiliar to me, which makes it even more interesting. Yeah, questions or comments? So you three are physically separated from each other, right? I mean, or Eli, you're at Washington?
Yeah, both Amy and me, we are at the University of Washington, and actually in the same department. And have joint positions in different departments, but yes, so we have same position at the Electrical and Computer Engineering department. Yep. And Maria is at Purdue.
At Purdue, I see. Were you had pre-existing collaboration, or is this sort of a new possibilities for you?
Yeah, we had a pre-existing collaboration, but this is definitely going to sort of strengthen it, and then add in, the hope would be to also add in, you know, the co-design, right, of the algorithms, with also thinking about hardware implementations, which will be ultimately really important for experiments.
And hi, can everyone hear me?
Yep.
Great. Thanks for your talk. It really helped me to understand deeper about a neuroscience project. In particle physics, we quite usually need to compare our collision data with simulation based on the state-of-the-art theory. In neuroscience field, are there equivalent approach, or is mostly really directly work on the data and try to understanding the neural data with the observed behavior?
Yeah, that's a good question, and somewhere where I think that neuroscience is at a very different stage as a field than physics, where the theories, I think right now the experiments and the data tend to lead a little bit more than the theories. There certainly is a lot of interesting work in theoretical neuroscience, and increasingly there's, particularly as people are digging more into, you know, things like recurrent neural networks and trying to understand principles of how they compute. There is more interaction between sort of theory and experiment, but by and large, I would say it's driven mostly by the data because we have relatively few. The theory is still evolving, I guess I'll put it that way, right? There's sort of not a set of, you know, expected outcomes, and then people are designing experiments to test those necessarily. But Ellie, I'd be curious if you see it a little differently.
Yeah, I mentioned modeling as one of these future prospects for the investigations, and we already model systems and kind of pretty excited about modeling full nervous systems, but these are at a much lower scale than the actual experimental systems that we can get data from. For example, like the modeling in our case would be of a very simple organism like C. elegans that has about 300 neurons. And in terms of recordings in primates or in mice, this would be on several orders of magnitude higher than that. So before we get this information, we cannot really set up a model that would be faithful enough to represent the nervous system.
Thank you. I have one other question related to the data collection. My understanding is that Maria is based on the optical approach, and Amy is more on electrical approach. In terms of the need of the real-time processing, comparing the two, are there different scale difference, or the challenges are similar, the required latency and also the required throughput bandwidth?
Yeah, that's a good question. I think the imaging data, and Maria can correct me if I'm wrong, but my understanding is it's definitely larger than the electrophysiology data in general. So the bandwidth for any real-time computation would probably be larger with optical. But then in terms of the timing requirements, to some extent it depends on the experiment you're trying to implement. But given that you were, if you were trying to implement the same experiment, for instance, that example I gave of recording in one area, brain area, and then stimulating another brain area based on the activity of this, the timing dependencies and requirements would be the same, independent of the modality there, because it's the timing is based on the biology that you induce plasticity by having a certain window of time between those two events in the brain.
Okay, so my understanding is that the latency requirement from the two approach are similar, but there's additional throughput challenge from the image method that Maria tried to pursue.
Yeah.
Okay.
Yeah,
Good.
yeah. And I think that, you know, in terms of the specifics of the proposal and what we initially scoped, I think that was part of where the optical approaches focus a little bit more on kind of high throughput, but not necessarily really low latency. And then the electrical, we kind of focused on the really low latency applications. But obviously, in the grand scheme of things, low latency applications for either modality would be really impactful for the neuroscience community.
Great, thank you.
All right, I think unless there are more questions, I think that's the end of the session. Yeah, thanks again. Those are really, really great talks. And I'm looking forward to understanding more. It sounds pretty cool.
Great. All right, thanks.
All right, we'll see everybody next week. I think the last one of these is next week, right? Shichia, is that right?
Yes.
Yep. All right. Bye.
Great. Bye, everyone.